Without further delay, I would like to dive in now into agenda item 4. And the first on my list is GBIF. Tim Hirsch is Deputy Director at the Global Biodiversity Information Facility and will present about the findings of the Global Biodiversity Outlook 5. So floor, microphone to you, Tim.
Thank you very much indeed, Chair. Can I just check that people are seeing my screen? I believe so. So yes, I'm going to talk a little bit, first of all, just to remind everyone, about the main conclusion within the Global Biodiversity Outlook that was published in September, GBO-5, regarding Target 19 on the sharing of information and knowledge, which was referred to by the Executive Secretary yesterday. And this was one of the few of the Aichi biodiversity targets for which there was partial achievement. And just a reminder of the overall wording of the conclusion of GBO-5 with regard to Target 19 is that although it found significant progress in the generation, sharing, and assessment of knowledge and data on biodiversity, there were significant gaps, major imbalances that remain in the location and taxonomic focus, both of studies and monitoring of biodiversity data, and also in the application of biodiversity knowledge in decision making. And that, I guess, represents one of the big challenges that lies ahead for our community in the coming 10 years. And you'll see just at the bottom of the screen there that one of the indicators of progress towards Target 19 was the mobilization of data through GBIF, the Global Biodiversity Information Facility. And I'll move on to talking a little bit about that now. So just briefly what GBIF is, we are an intergovernmental network and research infrastructure set up in 2001. With the aim of providing anyone anywhere with free and open access to biodiversity data. And you can see from some of the numbers just on our homepage there that our global community has mobilized currently more than 1.6 billion species occurrence records coming from more than 1,600 institutions worldwide. What GBIF facilitates is the bringing together of data representing evidence of many different kinds, very heterogeneous forms of evidence from, for example, natural history collections, from taxonomic collections, observations, also increasingly, for example, environmental DNA. And through the use of common standards, common informatics standards, sharing an informatics language, if you like, enabling all of that diverse evidence to be brought together into a single interface and many national and thematic portals, then for onward use into research and policy, including, of course, implementation of the CBD. In terms of what GBIF and that community, that worldwide effort is able to provide, it includes indicators of progress for various of the IHE targets, so Target 19 that I already mentioned, but also, for example, indicators on Target 9 on invasive species, Target 11 on protected areas, and Target 13 in terms of genetic diversity, for example, in the availability of data on socioeconomically and culturally valuable species. Capacity development has been a core part of GBIF's activity really from the beginning, but that has stepped up a lot in the last few years. And there's just this one program I would highlight, was mentioned yesterday, the Biodiversity Information for Development program, which has principal funding through the European Union. In the first phase of that program, we have been able to support 63 projects in sub-Saharan Africa, the Caribbean, and the Pacific, with a focus on mobilizing data from collections and other data networks through combined capacity enhancement workshops, which helps to train project teams and build up a community of practice in biodiversity informatics. One take-home from message from for example, the green circles on the screen there, is you can see that while we were able to fund 63 projects in those 3 regions, we had, when we put out the calls for proposals, we had more than 500 concept notes submitted. So what we're able to provide in terms of capacity support is far short of the demand. In terms of some of the outcomes of the first phase of this program, you can see there on the left some examples of the kind of projects that were supported and where just the data points were located in terms of sub-Saharan Africa. And crucially, again, I think an important message of this is that through this program, we're building up a pool of skilled people in the target countries and bringing together voluntary efforts in terms of mentoring and training from across our global community. An important point, I think, in terms of the benefits and the importance of mobilizing these collections, often even if they are not very large volumes of data, is the value that these data sets have in bringing new data, filling data gaps in the target countries. So for example, of 80,000 different species that were covered by the data mobilized through this program, 27% of them were species recorded for the first time in GBIF for a particular country. And that includes, for example, data on more than 1,000 threatened species, of which 22 were the first records anywhere in the world. So it shows the reach that can be leveraged from relatively modest sums of funding. Just briefly, a mention of another regional level capacity project that we've been able to
1 minute left.
Yes, thank you. That we've been able to implement through support from the Ministry of Environment in Japan, in Asia, and you can see examples of the projects there. And we have another call for proposals covering Southeast, Northeast, and South Asia coming up in December. And just a final reflection in this presentation, going back to GBO-5, GBO-5 outlined 8 areas of transition that were judged to be necessary to bring about the transformative change within the post-2020 framework. And I've just taken 4 examples there of these transitions on sustainable agriculture, fresh water, cities and infrastructure, and so-called Biodiversity One Health, and showing examples there of the kind of data for which taxonomic capacity is essential, that we really need to be able to implement these transitions. So soil biodiversity data, for example, for agriculture, freshwater species data and invasive alien species data, also for freshwater within urban biodiversity, citizen science initiatives, crucially important, and data from impact assessments for infrastructure, and finally for health, data on disease vectors, hosts and reservoirs, and, for example, medicinal plants data. So thank you very much indeed for your attention.
Thank you very much, GBIF. Thank you, Tim. For those who have joined, this is the second day of the GTI forum. Welcome, and we are under agenda item 4 and listening to interventions on achievements by parties and stakeholders in contribution to the GTI. As said before, we have a tight agenda, and if you bear with me, interventions should be restricted to up to 5 minutes, and I will, as just demonstrated, try to keep you in that time frame. Also, just as a pre-indication, we would intend to have a short break after 1 hour. Without further ado, let's continue with our contributions from participants. Next on my list is iBOL, the International Barcode of Life, and it's a great pleasure to welcome Donald Hoburn. for iBOL. Donald, of course, has an IT background and was with GBIF and ALA before, and is now representing iBOL and will talk to us about application of DNA barcoding and metabarcoding and conservation and sustainable use of biodiversity. Donald, please.
Thank you very much indeed, Christoph. Can you see my slides, by the way? So what I'm here today is I'm here as the International Barcode of Life Consortium's Executive Secretary, and I wanted to make a few comments on the place of DNA barcoding, metabarcoding, and in general sequencing technologies in support of the GTI. And I think it's important to emphasize up front that these are tools. They are not a complete solution, but much as the microscope did in an earlier age, they allow us to probe and explore species level biodiversity in ways that were not previously possible. It's quite clear that we have a massive need for large scale, repeatable information in order to support the global conservation, the strategy for conservation, and also to underpin the place of biodiversity within the Sustainable Development Goals, and for many other purposes as well. We live in a world in which we depend fundamentally on the biosphere to support everything that we do. And yet our ability to survey and understand and track change in the natural world is very limited. Historically, we've been relatively good at monitoring the state of vascular plants and birds and some other vertebrate groups, but much of life, including many of the organisms that interact with our food system or that are fundamental to the operation of ecosystems, are very difficult to survey. And part of that, the reason for that, a major part of the reason for that, is the challenge around the taxonomy and the identification of these organisms. And so the advent of increasingly cheap and increasingly flexible approaches to sequencing organisms is opening doors that allow us to rethink how we tackle these problems, and really to build a bridge between the ongoing work of taxonomy and describing and classifying the world's species on one side, and our need to be able to survey patterns and monitor the state and changes of biodiversity in the field on the other. We can do these two things in parallel and rely over time on mapping backwards and forwards between these two spheres using sequences as the connecting glue. The International Barcode of Life Consortium is committed to developing an Earth observation system for biodiversity. At present, the countries shown here in orange and in blue are members of the Barcode of Life Consortium. And in each case, there is an institution, an organization that is seeking to promote and expand the sequencing of these organisms as part of this Earth observation system. Fundamentally, we're talking about two paths for using DNA and the short stretch of DNA that forms the DNA barcode as part of our tool set. One of these, normally just referred to as barcoding, is taking individual specimens, gaining a sequence from each of these, and building up a library that helps us to recognize those same organisms again when we find the same sequence in the field. This can be a support for taxonomy by helping researchers to confirm the patterns that they've seen over the centuries through morphological study, but more importantly, it's a way to take all of their discoveries and turn them into tools that we can use more effectively for field-based identification. Metabarcoding is the taking of samples of organisms, either as here from a trap that has collected a lot of insects, or potentially just from soil and water and other parts of the environment and running it through the same sequencing systems to discover what sequences can be found. And this can be a very cheap and effective way for us to survey organisms such as insects and fungi, where otherwise the work of getting all of these organisms identified would be insurmountable. And it gives us usable information for recognizing repeated occurrences of organisms, even in those cases where we do not yet have a species name that we can put on the organism. There's a third path that the International Barcode of Life is also exploring, and that is the ability to take a single organism, such as a caterpillar on the left, and to run it through the sequencing process to find what other organisms are occupying it, bacteria and parasites, and also to understand, for example, in the case of this caterpillar, what food plants it's been eating. And so through an approach like this, it also gives us a chance to look at the ecology of organisms, which I believe would increasingly be a useful tool in conservation and in biosecurity and pest management. iBOL has just started a new 6-year mission. Well, it started last year, focused on massively upscaling what it has already achieved. We already have a reference library of millions of sequences from specimens, but large parts of the known biodiversity are still not labeled with sequences that we can detect in the field. At the same time, we want to be ramping up the amount that we are surveying ecoregions around the world to start setting up baselines and time series for the occurrence of different species, especially insects at this point in communities. And as I've mentioned, there's also a focus on using this whole process so that we can better understand species interactions. In his presentation just now, Tim Hirsch mentioned the diversity of sources of information that are contributing to GBIF, and through GBIF supporting the whole stack of international activities for understanding and protecting and responding to the needs of biodiversity. Sequencing organisms is increasingly going to be a tool that transforms how we go about this work, allowing us to probe and focus on those groups of organisms that were otherwise too difficult. Thank you.
Thank you very much, Donald, for this very concise and ambitious presentation. Just again, a reminder, since we are short of time for today and have little time for discussion, if you have questions to the interventions or to the presenters, put them in writing in the question box, and we will try to come back to you either if time allows at the end or in between from some of the presenters. Let's continue now with the next intervention, which is by CTAF, the Consortium of European Taxonomic Facilities. And it's my pleasure to give the floor to Professor Michelle Price. She is Head of Science and Curator at the Conservancy and Botanical Garden in Geneva, Switzerland. Michelle, the floor is yours.
Thank you very much, Christoph. So today I'm going to just give an example of a European consortium that's based on natural history collections institutions, which of course has taxonomy at their heart. So what I want to really show is how working together and how institutions pooling resources can really actually help to support progress in taxonomy. And it's very exciting to see the barcoding presentation before, because this is obviously opening up many new avenues of research for natural history institutions. So CTAF was actually formed in 1996, so it's exactly the same year in which the GTI was conceived of as well. So it has a long history of collaboration and cooperation across Europe. So it's got three, oh, sorry, next slide, please. Sorry, Katie, I totally forgot. So it works under three main objectives, basically promoting research, forming partnerships, and also becoming a political voice. And this is happening recently because CTAF has been able to influence the development of Horizon Europe, which is the new funding program for European Union. So next, please. Thank you. So CTAF is based on 6 strategic focus areas, which range from taxonomy training and e-learning, which is very important in supporting the GTI, right through to research and biodiversity informatics, which touches a little bit on the talk from GBIF and also from iBOL. The biodiversity informatics part is evolving because this part will become much more active under the DISCO initiative, which is a European research infrastructure that's just been integrated into the European roadmap. So CTAF has been involved in the development of DISCO, and this diversity informatics part is really something that DISCO will take on in the future. Next, please. So we realised that working together is actually a very successful model, and CTAF becomes more than the sum of its parts. Pooling institutions, knowledge, expertise, and all of the different types of researcher and collections manager in institutions is a very effective model for supporting taxonomy, supporting research, and also supporting collections. And collections, as seen in the presentation previously, form a big part of the Biodiversity Information Knowledge Network, some of the core data. So CTAF has partnerships with many other like organisations like GBIF and Spinach, DISCO, Biosys, GBIF, BHL, Catalogue of Life, and BGCI. And this is important because it means we can all align and we can all work together in cohesion towards answering different questions. And certainly now, with the Anthropocene challenge, we need to start addressing these relatively rapidly, or really rapidly rather than that. So next, please. So one of the current activities of CTAF is actually developing a research agenda for natural history museums and institutions in Europe. What this will do will actually provide the chance for us to identify questions that we really want to answer and also address emerging issues. And of course, in this, we can also align with future GTI action. So CTAF works in a sort of a hierarchical structure of the governing board. This is made up of the members, the executive committee, an elected body which oversees CTAF. And then the most important part of CTAF is their special interest groups, the working groups. Some really exciting initiatives have come out of CTAF, all been supported by CTAF. So we have the European Journal of Taxonomy for taxonomy publishing, and also CTAF is supporting the Distributed European School of Taxonomy, DEST, and other projects like BioTalent, which was looking at integrated ways to provide taxonomy training resources. So some online courses and then some field courses, and this was a really successful model that we can use in the future to offer taxonomy training in different contexts, especially now with COVID, when we're all almost all restricted at home. This possibility to actually give courses online is really something we have to explore. Taxonomy forms the foundation for ensuring biodiversity preservation and ecosystem sustainability. And with the exciting new developments in the meta-barcoding and the barcoding and all these possibilities this opens up, this means that collections institutions can play a very important role in underpinning these new discoveries. As Donald was saying, there's really a place for everyone to work together because we need to discover species, but we also need to document them for future generations. And this happens in the collections. Next, please. Just on taxonomy before, we've had several workshops building up to taxonomy, and we made a contribution to the GTI Forum in Egypt in 2018 with a taxonomy declaration. Next, please.
1 minute.
Thank you. We also worked jointly with GBIF to form action points, and these action points should become relevant in the discussions we'll be having today. Next, please. So we've actually shown, we mentioned yesterday, the CTAF DISCO International COVID Task Force. This is something that's very important, and we really see the importance of cross-disciplinary research in underpinning the things that we are talking about today, and will be talking about today in this workshop. So one of the important roles that we can play as collections and taxonomists is providing indicators based on scientific evidence for biodiversity monitoring. And I think this is another key theme that we will be addressing today in our discussions. Next, please. So again, one of the extremely important issues is going to be taxonomy training over the next years, building up a functioning taxonomy training structure, and also supporting the current existing taxonomy training schools. Next, please. So CTAF is both a contributor to and a stakeholder in science-based diversity knowledge. So I just hope this gives you a little example of how collaborating on an international level can really help mobilize resources, get experts together, pooling resources, and working towards understanding biodiversity in a much more fundamental way. Thank you very much.
Thank you very much, Michelle, and thanks to CTAF. And my apologies for being strict on time, but I really want to give everybody the opportunity to speak. Just a brief technical reminder to all presenters: if you're not using a headset, please make sure you speak close to your microphone and try to speak slowly, because the audio transmission is still a challenge under this system. Thank you. So let's continue now. Next intervention is by SANBI, the South African National Biodiversity Institute. And it's a great pleasure to welcome Professor Ramakbai Sebola from SANBI, who will talk to us about GTI achievements and further needs in South Africa. You have the floor.
Thank you. Right, Katie, can move on slides, please. Right, this is just an outline of what I'd like to touch on. Key milestones and some of the collaborations we're involved in, and some of the key challenges that we face at a national level. Next slide, please. Right, I think one of the key activities that have been occupying us for quite some time is the documentation of the South African flora, and here contributing to the online flora and electronic flora for South Africa, targeting about 20,000 species, over 20,000 species, and also an e-flora for South Africa, fauna for South Africa, looking at around 7,000 species. And the value add of this is that the current knowledge on all species and population diversity is pooled, and it allows us at a national level to determine priority species and setting of targets for conservation. Next slide, please. Right, for the past 10 years, we have concluded a human development capacity strategy, and with the horizon for 20 years, we've just now gone through the first 10 years, and up to 30 years, we're looking at mobilizing and skilling young people, and so far we have had almost around 1,000, close to 1,000 young people placed and trained, and about 44 national institutions participating in the implementation of this biodiversity human capitals strategy. And the value add is that it's not only one institution that helps to drive the development of capacity, but is pulling resources to invest in future biodiversity scientists, conservationists, and policymakers, using all resources sitting at various institutions. And we are also collectively able from time to time to determine critical and priority skills for the future. Next slide, please. And we have our NBSAP is Horizon 2025, and to date, there are about 55 targets that align with the IG targets, and 27% of those of the activities that are in the NBSAP have been fully achieved, 64% partially achieved, and 6% not achieved, and 3% having been assessed to be no longer applicable. And we also have to pull together various initiatives at a national level to mobilize biodiversity data. Key of those is the Foundational Biodiversity Information Program. Here we fund the mobilization of foundational data around species descriptions and the occurrence data. And we also invest in improving the research infrastructure for biodiversity collections, digitizing and storing and making sure that information is shared among national institutions. And we also do a similar initiative on the genetic material. National Biobank have been networked fully, and we are also moving to strengthen the capacity for the country to be able to share material across. Next slide, please. We also do regular assessments on status of biodiversity. We have National Biodiversity Assessment. The status here is to look at the status of South Africa's ecosystems and biodiversity. We have just released one last year, NBA for South Africa, and it covers all the ecosystems and covering populations, and also the status of biological invasions and their management in South Africa. These are the two key assessments that we have been able to achieve, and it involves a network of various institutions in the country and elsewhere. And the value add is that current knowledge on species and populations put together, and it allows also for determination of priority species and setting of targets for conservation, synthesis of best available science on ecosystems, species, and genetic diversity across all realms. We cover a whole range of from terrestrial, inland, aquatic, estuarine, marine, as well as subantarctic territory as the focus of these assessments. And out of this, the latest achievement has been able for South Africa to complete the Rate List Index for different taxonomic. There's also efforts to link up with our region, within our region, and also at the continent-wide as well as global in efforts for conservation. Just pick up a few of those here is the transfrontier parks and conservation areas that have been demarcated across South Africa with our neighboring countries. And we also participate in the Millennium Seed Bank partnership with Q and the United Kingdom, where collections of seeds from endemic and threatened species is a major activity for ex-situ conservation. We also are involved in the crop relative, and here there's also partnerships with Mauritius and Zambia. And we're also having to participate in the AU process towards the end of 2063. Next slide, please. Yeah, this slide I'll just skip this one here. This is talking to just to show you some of those conservation areas in the region. There's about 6 of those that cover 6 countries that are bordering South Africa. Next slide, please. And we also contribute very largely to GBIF and also part of participating in the IB-based assessments. Next slide, please. Here's just to share with you the level of contribution from South African biodiversity data to GBIF platform. This is a range of areas from animals, plants, fungi, and so forth that we contribute. I just want to end up with the highlight of a few challenges that we experienced. Next slide, please. We often find that data is still, the data sharing is poor, and decentralized national biodiversity databases hampering that data sharing. Ineffective coordination and biodiversity data generation, part of it, as well as collection across the continent. There's also inadequate resources that are needed in terms of finances and human capacity for implementation of our national strategies and plans, and lack of access to appropriate technologies in some areas that we need to focus our attention to going forward. I'd like to thank you very much.
Thank you very much, Ramakwai, and thanks for this impressive list of activities and still remaining challenges. We continue with the Royal Belgian Institute of Natural Sciences. Dr. Jolien Venneman is a science program officer at Arbens in Brussels, and she will talk to us about good practices and recommendations from the Belgian GTI National Focal Point. Jolien, please.
Yes, hello. Good morning, afternoon, everyone. Thank you, Chair, for the introduction and for the opportunity to take the floor. I'm actually the new Belgian focal point to the GTI. In this presentation, I would just like to give a very brief overview of some of our good practices, as well as some of our hopes and wishes for the post-2020 framework. My predecessor was Marie-Lucie Suzini. She was the Belgian GTI focal point during the past 10 years. practices I will be presenting are actually the result of her efforts. So the Belgian GTI focal point is actually part of the CBIOS program, which is a part of the Royal Belgian Institute of Natural Sciences. It stands for Capacities for Biodiversity and Sustainable Development, and it's funded by the Belgian Development Corporation. So CBIOS aims to strengthen the capacities of our partners in the South, mainly African partners, and this in the field of biodiversity conservation and sustainable management. And actually the GTI part, or the strengthening of the taxonomic capacities of our partners, is quite an important and dedicated part of the CBIOS program, as we have several key recurring GTI-related activities, among which the provision of research grants to young taxonomists from the South, to enable them to follow a taxonomic training in Belgium, and to access our Belgian collections during a period of 4 weeks. You're actually
Sorry, Julianne, you're self-muted. We lost your voice.
Yeah, I'm back again, I think.
Thank you.
Yeah, now we can hear you. Please continue.
So I was saying that we are organizing one competitive call every year. And as you can see on the map, this has been very successful. So since 2004, we have supported over 200 internships for 128 grantees coming from 33 different countries. And as you can see on the diagram, we support the work on several taxonomic groups, but it appears that insects have been studied most frequently by our grantees over the years. Another call we are organizing every year is actually addressed to our Belgian researchers, mostly the researchers from our own institute, who want to organize a taxonomic training in the partner country itself. So far, we have organized 62 of such projects in 15 different countries, which has resulted in some very interesting collaborations and also outputs, among which a description of new species. Thirdly, we are supporting the publication of ABC Taxa, which is a series of peer-reviewed manuals that aims to strengthen the knowledge transfer in the field of taxonomy and collection management. All of our volumes, including the latest one, can be downloaded for free on the ABC Taxa website. And we are also distributing hard copies, which are always available on request. As far as our awareness raising activities are concerned, every few years we organize a specific workshop for our GTI alumni. And we also provide grants through competitive calls to support their awareness projects in their country. And then to support the science policy interface, we help them in writing policy briefs, which are aimed at decision makers in their countries. And ideally, such a document is the result of an uptake meeting that is organized in the partner country itself to inform the local decision makers and stakeholders about the taxonomic results of a project. And actually, the policy brief on insect biodiversity in Vietnam that is shown on the slide was the result of such an uptake meeting. This was very successful, so we definitely plan to continue this in the future. And then finally, I would like to conclude with some aspects which we hope will be considered or will receive some special attention in the post-2020 framework. I will not discuss them all because some of them have already been covered and I'm limited in time. But for now, I would like to say that we consider point 1, so the sustainability of capacity development outcomes and funding, as quite an important one, as well as number 3, the easy access to and the generation and use of digital information, and more specifically digital sequence information, which is quite a hot topic right now. And of course, DSA is also linked to number 4, the advances in molecular techniques such as DNA barcoding, which has already been covered by some of you. And I just want to say that starting from next year, we are actually planning to organize at least one workshop a year in our partner countries that covers DNA barcoding and bioinformatics. So I know that some of you have already done that. So we are always open for collaboration to make joint training material and courses. Because in line with what Tim Hirsch said yesterday, we are also very much in favor of pooling and sharing knowledge and resources and experience. So thank you very much.
Thank you very much, Julianne, for this, again, impressive report from Belgium, and also especially for the outlook, what we could or should do in the future. I would like to continue, but let me check with the Secretariat. Is Dan Jansen with us? I don't see him on my list.
Yes, he is an attendee. He's just sharing a screen now.
Okay, great. So we can continue with a contribution, so to speak, from Costa Rica. Then Professor Dan Jansen, of course, is well known and long-time evolutionary ecologist who spent most of his career building and advising conservation action for specifically the Guadalcanal conservation area in Costa Rica. And he will talk to us about BioAlfa, Bio-Literate Conservationist National, Wild Biodevelopment. Dan, please.
Good morning, everybody. Thank you for allowing us on the stage here for a moment. And thanks to Junko and Katie for that. We are effectively science reporters reporting on what's going on in Costa Rica. The major thing that's relevant here is this project called BioAlfa, which is Spanish for bioliterate. The philosophy behind this is that conservation itself is national wild biodevelopment. It's to create the bioliteracy platform of Costa Rica's millions of species for the use by its 5 million people. It's a national deep dive through what you heard from Paul and from Don Hobart about Bioscan's global biodiversity. So what it really is to understand what a species is, what a species is with a name, where it still survives, what it does, and how to find it. And put that platform on the public web. So the day coming up very soon when everybody, all of you and everybody in Costa Rica and Africa will have in their back pocket a personal, very cheap, reusable barcoder that actually captures the barcode and allows us then to know everything we know about it, whatever we know and is on the web. This is about the national capital and the natural capital of Costa Rica in a self-sustaining garden. And there's more than 1 million species of it, not counting microbes, sitting there in this little tiny country. The goal is to integrate this natural capital in the socioeconomics of the country. And to do that, we need to know what it is, where it is, what it does, and get that in the public domain. This is a three-way crop interview. This is one crop here. These are 5th graders from farm children. This is one crop here, a non-poisonous snake that this child is showing to these children. And the third crop are these electronic communications with the world. Every time one of these people takes a photograph of this interview, that means that 40 or 50 or 100 or whatever number of people are seeing the same thing. Now, what is the information itself? So Ruth here, 16 years old, notices caterpillar turds on the ground. She asks Memo to go up to get the ladder. She climbs the ladder, finds the thing that was generating the turds, and there's the caterpillar that was dropping the turds. They go in a plastic bag with their food. They become reared. They get database. They get photographed. And all of that goes on the web. These people never made it past 6th to 7th grade. They have no university degrees. They're plucked out of the agricultural countryside and treated like good graduate students. And they learn and they do, and they've been doing so for now for 35 years. They also write up this stuff. These are the bugs they know. There's more than 1,000 of these on the web right now. And behind each one of these logos, there are paragraphs of information. And behind that particular one right there, this is the author 33 years ago. Never made it through 6th grade, had never written a full sentence in his life, and certainly had never put together a paragraph with an image. And yet he produced this document. Oops, sorry. He produced this document right there behind that, 2 pages of caterpillar photographs and text to go with it, which is being looked at from Chile to northern Mexico. It's also in Spanish, of course. Now that's the bottom level. This is the top level. This is Costa Rica's president right here, talking about BioAlfa to the cabinet. and signing a decree which states that this information and the barcode are public domain, meaning they are freely shared with the whole world, as well as, of course, all of Costa Rica. This is the only tropical country in the world that has formally declared that a piece of its genetic information is available to everybody. That's part of Costa Rica trying to be a green country that integrates its greenness in the rest of the society. We all know what that is. Each barcode is a one-word blog of 658 letters in a 4-letter alphabet. You can explain that to any 5th grader. There are more than 1 million of them in Costa Rica, and it's unique for every one of those species, bar about 10 individuals. So this is the actual barcode for this actual fly right here, which is reared, so we know what its caterpillar host was. We know now what it is, and we can connect it to whatever new species name has just been given to it. And I say new species name because of those million species, not more than 5% have got names. We are dealing with a completely unknown universe. This is not like Central Europe. So how do you do that? You trap them in all kinds of ways. This is a very common one called a malaise trap. The insects fly into it, they end up in a bottle of alcohol, and you change the alcohol every week. That takes no previous knowledge to do that.
1 minute.
So let's show you what it looks like. Here's the conservation area itself, the size of Philadelphia. You put one trap there, one trap there, and one trap there, 17 kilometers apart. Run them per year. You get 17,000 species. We now have 22,000 species from one single trap. Easily, 95% of those species have never been described. And of these 3 pools from 3 different malaise traps, out of 17,000, only 31 occur in common. That tells you the size of the challenge. That challenge is now spread over the whole country. Every one of these yellow stars and the red stars are 2 malaise traps run by the park system on their budgets, on their salaries, on their time, who know nothing about biology. But they're learning by running a malaise trap and catching insects. Right now, that's 200,000 specimens sitting in Guelph to be barcoded. And that is how these people are going to learn. They are going to do the inventory of the country. So the last slide here, we now know it's nationally feasible for a biodiverse tropical country with its international collaborations to generate fine-grained taxonomic knowledge for a massive complex biodiversity and its collateral, and put that on the web. There is a national pragmatic strategy in hand. The technologies for this, the social involvement for this, and all the puzzles can go around and absorb the taxonomic impediment. No problem. Takes labor, takes money, takes time, but it can go around the impediment. This going around is essential if taxonomic product is to reach its highest and best uses, which is to facilitate a sustainable wild biodiversity that's integrated with its human societies, and that should spread like a virus from country to country. Thank you.
Thanks very much, Dan, for that impressive program and good outlook from Costa Rica. We continue straight and move to the Bahamas now. It's my great pleasure to welcome Dr. Ethan Freyd. He's a botanist at the Leon Levy Native Plant Preserve in the Bahamas and will introduce us to the GTI in the Bahamas. Ethan, please.
So good morning, good afternoon, and good evening. Thank you for this opportunity to talk about the global taxonomic initiative work going on in the Bahamas. The Bahamas is occurs west of Florida and north of Cuba and Hispaniola. It's a low-lying set of limestone islands covering more than 6 degrees of latitude, straddling the Tropic of Cancer. The country is primarily a marine environment with slivers of limestone ridges running along edges of shallow banks with very deep water canyons. The biodiversity of the Bahamas has been studied for a long time, and it is relatively well known. Now, given the short time slot, I'm only going to focus on a small slice of all the work currently underway that applies to the Global Taxonomic Initiative. This work is primarily being undertaken by the Bahamas National Trust, which runs the national park system here. In the last decade, there's been a robust and ongoing plant awareness and education campaign. This campaign started in 2011 with the development of the Leon Levy Native Plant Preserve. The 12-hectare site is focused entirely on plant conservation, education, and outreach. And it is an internationally accredited botanical garden. In 2014, the Bahamas National Trust developed a science and conservation program at the Levy Preserve using the Global Strategy for Plant Conservation as its backbone. At the Levy Preserve, there's progress on 9 of the 16 targets, and within the larger National Trust organization on another 4 targets. Most of the Global Strategy for Plant Conservation targets apply to the GTI. In our living collection now, there's 1/3 of all the Bahamas total plant species and 50% of the Bahamas endemic plants in display beds with labels and available for taxonomic training. At the Levy Preserve website, there's an online flora with 50% of the country's diversity with detailed descriptions and imagery, and it is integrated with the World Flora Online. There is now a native plant nursery that has over 85 species of trees and shrubs, and this has allowed the development of what we call our Adopt a Species program for the local school systems, with each school having a signature plant species associated, and then the students learning about those species. And we as well as have what's called a Forest on a Pallet program that sends pallets of trees to other islands for restoration and education. There's a collaboration with the National Art Gallery of the Bahamas to create a mostly native tree sculpture garden forest. This is on a half-hectare site next to the gallery in the downtown area of the capital. The art gallery has over 100,000 visitors a year, including all of the school children on the island. So it started looking like this, and then over a 2-year period, 500 trees were brought in, transforming a highly degraded environment into a forest in the city with plant identification signs. This is what it looks like today. In 2019, we had the publication of a terrestrial field guide that covers botany, ornithology, herpetology, entomology, mycology, and geology. In 2019, the Postal Service published a series of stamps that highlighted endemic plant species, and in 2020 published another set that focused on medicinal plants. In 2014, a formal training program began for Bahamian University level students. It trains field biologists in botany, ornithology, herpetology, and geology. To date, we've had 17 students go through the program, with many of them now working in the environmental industry and within government. The Bahamas continues to work on the development of additional marine and terrestrial parks and building in-country natural capacity. Progress is being made on almost all activities of the Taxonomic Initiative for both plants and animals, but I would need much more time to fully discuss all of this. Thank you.
Continue, and the next on our list is Thomas Borsch. He's the director of the Botanical Garden and Botanical Museum of the Free University in Berlin, Germany, and of course an eminent plant systematist and taxonomist. And he will talk to us about the World Flora Online. Thomas, the floor is yours.
Thank you very much. Yeah, I'm here as one of the co-chairs of the Taxonomic Working Group of the World Flora Online. And well, I see 2 colleagues, Heining and Mark, who are also in this working group. And in the next slide, we see the portal. So the World Flora Online is a global initiative that includes right now 43 different institutions across the globe. You see all the logos, and it's basically supporting the global strategy for plant conservation. Next, please. So the target 1 of this global strategy for plant conservation was adopted by the CBD in 2010 is the World Flora Online. And what is interesting here, and maybe this is can be taken also as a model for a group of organisms, land plants, that it was then subsequently implemented and led by the botanical science community. So plants are essential as primary producers. It's sort of organisms that we actively manage more than perhaps insects, so they have a specific relevance. And what this is about is providing quality information on plant biodiversity and not just names. So delimitation of species as fundamental biological unit is very important. And here we have this basically as a synthesis of scientific knowledge throughout by this global World Flora Online consortium, which works through a council with also very transparent decision-making procedures. I think this is also very relevant in order to how to organize this workflow. Next, please. This is, I think, something very fundamental. We have to recognize and appreciate that taxonomic has started, well, basically with Linnaeus as species plantarum in the 1750s. But now it's in a strong transition to become a competitive science in the context of evolutionary biology. And I think this has strong impacts. Here we see the details don't matter really on the left-hand side. What could be a workflow starting from the specimens and collections leading to, well, species delimitation and then a name and a classification. And this is also very relevant because these workflows using fair data standards and current principles of evolutionary biology are currently redesigned within, for example, the plant life people and the World Flora Online context in an international way. And here we have the Mexican Instituto de Biología at Mexican National University, who is very involved in this procedure with a workshop Caryophyllalis, one lineage of flowering plants using as an exemplar. And what we really have seen is that it's not when you talk about taxonomic impediments, simply the question of, well, having access to technology, but it can be that there is excellent research done in a so-called developing country. It can also be there is lack of capacity in a so-called industrialized country. So it's really a mosaic of advances, and this has to be considered really coming from a scientific point of view. Next, please. How does the World Flora Online work? Well, what is very well known here in this orange box, regional treatments, Flora. So the plant people have been very intensely collaborating for decades, centuries, and it's regional treatments. But on the other hand, what we see in this, well, realm of evolutionary biology becoming more eminent, that we basically need taxonomic expert networks adopting specific groups and working together internationally really to channel the knowledge and also to bridge from active research to really synthesizing and delivering, for example, here in the World Flora Online portal. The next slide, please. We have, of course, thought about in the context of GTI what has facilitated the buildup of this taxonomic resource and global partnership. So this is the scientific community spirit to begin with, a spirit of collaboration and sharing resources. Then it's digitization of type specimens. JSTOR Global Plants has been very efficient worldwide in all herbaria to digitize and make available this information of type material in collections. The same to literature. BHL is also for other groups of organisms. Then it's the access to research data and specimens. This is very important, but also capacity building intertwined with joint research projects. And I really would like to stress this is not, when we look at the various projects going on in many institutions throughout the world, this is really something which is a mosaic-like progress step where capacity building can also mean needs to learn specific methods in a country like Germany, not only in other parts of the world. So this is really something that has to be addressed with really care and looking at how science works in the 21st century. Next slide, please. What is needed to be successful? Making it really short. The World Flora Online is intended to provide an authoritative, accurate, and comprehensive taxonomic information to guide biodiversity conservation and sustainable use. So it's not just names, it's knowing about understanding the species, their species limits, and if we think about a biodiversity strategy, also things like intraspecific diversity matters a lot when it comes, for example, to sustainably using plants as a natural resource. So the World Flora Online needs to continuously integrate discoveries in scientific progress. So I think this is very important as a philosophy. Science is approximation, and it will continue to deliver progress worldwide. Second, it wants to be a valuable ongoing contribution to a further phase of the global strategy of plant conservation. And we think this is target one of this. This was really well achieved in global partnership. And of course, we hope that it will be adopted and incorporated as part of the Global Biodiversity Framework 2030. And of course, parties to the convention need to continue to invest and support taxonomists. And I think what we all need to improve is linking taxonomists and conservationists and policymakers. And this is really relevant because it's a science. There are needs of a scientific community thinking, for example, in young scientists building their careers, but also from the user communities with their needs. And we think that's very important to bridge this very openly in terms, for example, of a clearinghouse mechanism to really advance for the interests of both domains. Thank you very much. And the next, thank you very much.
Thank you, Thomas, for bringing this very important program update to us. I've been informed that we now are really happy to welcome Diana Hernandez from Conabio. The connection seems to be there. And I'm happy to give the floor to Diana for her intervention on the species catalog in Mexico.
Yes, can you listen to me now?
Yes, we hear you well. Go ahead.
I'm happy. Thank you very much. Well, I'm going to talk about a very quick view of some of the programs that we have to address some of the targets of taxonomy and biodiversity knowledge through the National Biodiversity Information System. Next slide, please. Next, yes. First of all, well, to consider how many species are there in Mexico, and we have an estimation that we have among 10 to 12% of the world biodiversity, which means 200,000 species. Although there have been several approaches in trying to record all the species that have been recorded in Mexico, and this varies from 94,000 to 100,000 species. Next slide, please. Having that in mind, the mission of Conabio is to carry out different activities aimed to increase the knowledge on Mexico's biological diversity and its conservation and sustainable use. This can be done through the National System on Biodiversity, which is among the main activities of Conabio to instrument this system. Next slide, please. The system has 4 main components, and the core of this system is the information, the taxonomic information of a species through specimen occurrences and catalog of a species and species data profiles. In the next slide, please. Currently, we have more than 14 million of occurrences recorded in the system. That comes from 900 databases and projects that have been funded mainly by Conabio. And the next slide, please. All this occurrence represent more than 85,000 species, valid species from these different groups, different taxonomic groups. And this information is validated taxonomically and nomenclatural through the catalog of species. Next slide, please. The catalog constitutes the most complete and reliable system of taxonomic information species, and we used it also to strengthen our link with national and foreign taxonomists and promote their active participation. Next, please. Currently, we have included in this catalog 108 1,000 valid species. In the orange line, we can see the total increase of a species, and the other lines shows the different taxonomic groups. It's only valid species, and this has been achieved with a contribution of more than 160 taxonomists, mainly Mexican taxonomists from other institutions, through different projects and postdocs. Next slide, please. Another program that we have is the assessing program of the species, mainly focused on the assessment of the species that are already recorded in the national red list. And this program aims to complete the information, whole information of the species that are on risk already, or to assess another species or another priority species. Next slide, please. Another program is the System of Information on Invasive Species, which has gathered information of more than 2,000 species right now, with 600,000 occurrences. And this system has an annotated checklist of alien invasive species, which main objective is to identify major risk species for the country and to participate in national and regional initiatives. And the other program that I wanted to talk is about the catalog of collections and institutions that we have in SNP. We have gathered information of 800 collections. This program is available online, and each curator or taxonomist can update information directly on the page, on the web page, in order to keep updated this catalog and to be accessible for everybody. All the information or more information, you can find it in the link that I put on the bottom of each of the slides. So this was just a quick view of the programs. Next, please. Thank you very much.
Thank you very much. I understand that we are now ready to give the floor to Dr. Xin. His microphone seems to be working, so very happy to welcome Professor Xin from Institute of Botany, Academy of Sciences in Beijing. He will talk to us, as announced before, about GTI contributions from China.
Hello.
Yes, we hear you.
I would like to introduce what taxonomy community in China support the past GSPC works and what is the major plan for the future support the post 2020 biodiversity targets. Please. Okay, this is the overview of the different country GSPC progress that is made in the meeting 2 years ago in South Africa, Cape Town. And you can see that China is one of the top country that more progress to implementation of the target of GSPC. And you can see target 1, target 2, and target 3, and the other almost done 2 years ago. And last year's the Chinese government have made a assessment of the implementation of GSPC in China, and the situation is some similar to this table. Yeah, so it means that in the past more than 10 years, taxonomy community in China, particularly in the Chinese Academy of Science, they have more and more stronger support the implementation of the national target. Next one. And what is we doing in the past? I can briefly introduce to you. In the first aspect, that for taxonomy, we continually to collect the specimen in the something least contact correct area and more focusing on the some important taxa. And in the national level, every year the Chinese Academy of Science will be organized taxonomy to work on the catalog of China, and every year we will launch the annual checklist. And it will be China will be one of the few countries in the world that have a continual annual checklist. Other aspect that in the past 7 years for the Chinese government to make assessment of the red list of China plant and some important animal, and last year also complete to make the assessment red list of macrofungi, and all these red list assessment are majorly based on the taxonomy works, and many lot of taxonomy are involved these several red listed works. The other thing is we in the country's level we continue to determine the whole country specimen for plant. For the whole country, we have almost 16 million specimen of plant. So far, is 2 of a third is already determined, and actually the remaining 1/3 plant specimen is very poor, and is the data deficient. So the major plant specimen is already determined, and now they are available. Their data is available on different websites. So it's a electronic specimen information is useful for the country level, for education, for scientists, for conservation, and so on. And this is the time to show you what is the common idea is from my taxonomy community, and also improved by government, is what we should focus on in the next periods to give a more stronger for implementation or achievement of the biodiversity target. The one is we suggestion the government, appeal the government should set up a national and authority information center, because now is many institutions in China, even the different institutions of a Chinese Academy of Science, they are working on taxonomy conservation, but many of them is something is not share the information. So next, the first should be set up a centralized information center.
1 minute.
And the second one is we must pay more attention to yield the information from taxonomy and assessment of threatened species to using in the planning on biodiversity conservation. Even now, we already provide many taxonomy and red list information on the country level to make assessment about the gap of national park and so on, but we think it's not enough to use it. And Okay, next one. And also we, the taxonomy community now more and more involve the education or the activity of the civil societies. For instance, every year, different institutions of a Chinese chemical science, they will have a running different kind of a call for taxonomy and for my assessment of some threatened species, or even for deadline of specimen. So every year, at least they have a several kind of symposium or training call on the improve the taxonomy working or something like that. Yeah. And actually, more and more taxonomies have a suggestion or have a common idea to ask the state to give more strong support for taxonomy. For instance, last week we have a annual systematics and evolutionary conference, and the 14th young systematical conference, and it have 500 participants, and other 200 is attend in the web, and they have several common appeal for the state. The first one is appeal the government to open more taxonomy position to provide more funding for taxonomy, and the other one is to support the taxonomy journal, the journal periodical, more focus on publishing the taxonomy paper. Yeah.
Excuse me, could you please conclude?
And the other one is not only for the taxonomy community, but also the government ask the Chinese taxonomy should make close and more cooperation with the other countries' taxonomy or even international. For instance, as I know, for Chinese Academy of Science, they have a several cooperation with the neighboring country. For instance, in South Asia, something like Myanmar, we have a close relationship, joint program to look at the biodiversity, and the other one is in east of Africa. And personally and my group attend the East Asian networks. The name is East Asia Biodiversity Conservation Network. That is financial support by Korean National Arboretum, and this year is the star for second place. It look at the catalog of East Asia. The second one is made the assessment of threatened species common to this area. The third one is share the information, the specimen, and the photo on website. So there is encourage the scientists to make cooperation, not only in the country, but also to neighboring country.
Thank you.
The final one is, sorry. The
yeah,
final
could
one
you please conclude? The time allocated is really used up, and we have 3 more presentations to go.
Okay, the next one, please. Finish. Yeah, thank you. Thank you for your attention. Yeah.
Thank you very much, and apologies for being so strict on time and also for the technical challenges. Thank you very much, Heinin, and for this good update on GSPC and GTI related things from China. I've been informed that due to the time shortage, I should switch the sequence of speakers, and I'm now happy to give the floor to Dr. Hugo de Boer from Natural History Museum and University of Oslo in Norway. Hugo, please.
Thank you. Well, I want to thank you for inviting me, and I want to thank the Norwegian Ministry of Climate and Environment for nominating me as an expert. I'm here to talk a bit about what Norway has been doing with regards to the GTI. We've been training a new generation of biosystematists to meet society's needs for biodiversity expertise, and I'm here as a professor from the University of Oslo, but also as the leader of a research school in biosystematics and a member of the Norwegian Scientific Committee for Food and Environment. Go to the next slide. I'll try to stick to my time. In line with the GTI capacity building strategy, Norway established a Norwegian Biodiversity Information Center in 2009 with its own dedicated Norwegian Taxonomy initiative, as well as the Scandinavian Research School in Biosystematics in 2010. These two initiatives, they have yielded a lot of new species for Norway, a lot of new species new to science. Also, the research school has been providing courses in taxonomy and systematics. The last 10 years, we've taught about 100 courses to 1,000 students, and all of this is for free. And this has been very much a network that has encouraged people to seek a career in biosystematics. Next slide. So these initiatives, they have, of course, generated further capacity building. They have helped transfer taxonomic knowledge, intergenerational. It's also helped establish Norwegian participation in international networks such as iBOL, already mentioned, also Bioscan, already mentioned, GBIF, already discussed. Also DEST, the CTAF School of Distributed European School of Taxonomy, as well as, of course, EU projects, because if you're investing in science, it leads to more science. Next slide. So for the next 10 years, I envision that in Norway, we'll continue to progress beyond pure taxonomy capacity building. The Norwegian Environment Agency has taken the initiative in recent years to commission a number of reports on the use of environmental DNA and meta-barcoding and metagenomic approaches. And these, of course, are important because environmental DNA and meta-barcoding and metagenomics, they put a value on taxonomic knowledge because they stimulate a wider adoption of molecular biodiversity assessments in environmental monitoring. It reinforces the need for taxonomic expertise, because if we want to execute national laws on biodiversity, we need to have species knowledge. But importantly, the Norwegian Environment Agency, by initiating this work on environmental DNA, they are stimulating integration of eDNA methods into national law, and they're encouraging coordination of public and private initiatives. So we see a blossoming of consultancy firms, of semi-state organizations that are doing more and more biodiversity assessments based on DNA sequence data, for which they need taxonomic expertise. So a lot of our PhD students, they are not continuing in science. They are moving into private industry. And that, I think, is something that I would also like to see GTI look at. Not only how can we get more money for taxonomy, but how can we create and increase the value of taxonomy. That's what I wanted to share.
Thank you very much, Hugo, and thanks not only for staying well in time, but also for these very good ideas. We can continue now with the original sequence, and I'm personally certainly very happy to be able to return to Asia, and now to kindly invite An Ta. She is working as a specialist in the Nature and Biodiversity Conservation Agency under the Ministry of Natural Resources and Environment in Vietnam, in Hanoi. So, An Ta, you have the floor, please.
Thank you, Mr. Chair. And good evening, everybody. I will give my presentation short and simple. And I will talk about the what Vietnam done with the taxonomy initiative to implement the RG targets. Next, please. And first of all, as a national focal point for biodiversity in Vietnam, we developed the National Biodiversity Database System called NBDS, and we developed the NBDS with the idea of connecting with the GBIF and basing on Dark Core Foundation. So also for the taxonomy tree, we adapt from the catalog of life so that we can have a good connection with the global with the GBIF. And first of all, we do a pilot at one national park, and now we would like to expand the National Biodiversity Database System for the next 164 national park and protected area in Vietnam. And at the moment, we have a database of 30 national parks in Vietnam. Next, please. And also, we developed the national database system for genetic research, and we also basing on Dark Core Foundation, and we also first we make a pilot for one province in Vietnam, and now we will expand the system for other 62 provinces in the country. Next, please. And also, we are now continue carry out activity to build a database system on taxonomy and distribution of a number of endemic, endangered, and rare terrestrial vertebrates. And we also working on the plant species and also for the insect species. This activity is carried out at the Institute of Ecology and Biological Research in Vietnam. Thank you for your attention.
Much, An Ta, and for that overview about relevant activities in Vietnam. It's certainly very good to hear that. We can now move to the last speaker on my list for today, at least for under this item, and turn to the UK. I'm very pleased to offer the floor to Dr. Mark Watson, who is a senior scientist at the Royal Botanic Garden, Edinburgh, and he will talk to us about GTI experiences from the UK. Mark, you have the microphone. Please.
Thank you very much, and hello everyone, and thank you, Chair, for the opportunity to share some of the work of the UK institutions in support of the Global Taxonomic Initiative. You'll be pleased to know I've got just 3 slides focusing on topics of capacity building, advances in technology, and the sharing of taxonomic knowledge. I'm only going to be using examples from my own organisation's work in Nepal, but I'll try and include some of the work of the other organisations in the UK on non-plant groups. Next slide. Royal Botanic Garden Edinburgh has been working with partners in Nepal for nearly 20 years, initially addressing a major floristic knowledge gap. There's a checklist, but there was no flora. This pink map produced by Volterlac in Berlin in 2010 shows Nepal as a white as a conspicuous knowledge gap in Asia alongside Myanmar. But at that time in Nepal was also facing a generation gap in taxonomists. So we had to embark on a program of capacity building to train future generations. Many of these capacity building activities were informal, such as workshops and on-the-job training, if you like. But importantly, they also included postgraduate qualifications both in Nepal and the UK. Our experience is that capacity building really has to be long-term, and you need to provide continued support and career development in order to maintain and develop those skills that you provide. So we've done this through co-authorship on scientific papers, books, and reports, plus also co-working and co-research. And this really cements the partnerships and builds trust and understanding. Although I've been talking about UK-Nepal bilateral relationships, really it's important to say that many of these are multilateral, and particularly we work closely with partners in Japanese organisations. I'm pleased to say that our long-term partnership with Nepal has also encouraged and facilitated other taxonomic experts from many other countries. They come to Nepal and share their skills, and so continue the capacity building. Next slide, please. Advances in biodiversity informatics have really transformed the way that we undertake taxonomic research and how we make our data available. With the Flora Nepal project, we believe that's the first born digital flora, in that the primary taxonomic product is not the printed flora, but the database from which the volumes of the flora are published. And that database is also used to generate other taxonomic outputs via the web and via apps. This has enabled us to contribute to the World Flora Online. And you've heard about that from Thomas Borsch and also Ethan. This is a global partnership which really seeks to provide unified access to all this floristic data and view it in a global framework curated by taxonomic experts. And in UK institutes, we strongly support the World Flora Online, and we've been aligning our taxonomic research programs so they can contribute towards this vital international resource. Looking at other wider approaches, the DISCO project was also mentioned earlier, and this illustrates it's a good example of a regional approach to shared research infrastructure, and it brings together, or aims to bring together, all of the European natural science collections. This EU-funded project brings together EU-funded project aims to digitally unify all these collections under a common curation model, making the data easily available, findable, accessible, interoperable, and reusable. Thomas talked about the FAIR principle earlier. And at the UK national level, the Darwin Tree of Life is a new platform bringing together biodiversity, genomics and analysis partners to sequence the genomes of all 60,000 species of eukaryotic organisms in Britain and Ireland. The Darwin Tree of Life aims to transform the way we do biology, conservation, biology, biotechnology, very much the ambitions of the Bioscan project, which you've also heard of. Next slide, please. And the growing use of digital technologies allows us to mobilize data and greatly facilitates the sharing of this knowledge. And just taking 3 stakeholders as examples, researchers, wherever they're working, they can access primary data through institutional websites or international aggregators such as GBIF, and we've heard a lot about GBIF. GBIF is very important to the UK institutions. Over 192 data providers in the UK have provided over 113 million occurrence records to GBIF, covering all parts of the globe. And last year, the UK was the 2nd largest country level publisher of data to GBIF and the 3rd largest publisher of peer-reviewed articles using GBIF mediated data. The synthesized knowledge encapsulated in the Catalogue of Life, publications such as CITES checklists and other CITES guides are used by national policymakers in conservation planning and legislation, and UK institutions are active in producing these. We also feed data into countries developing their own action plans and reporting to the CBD. Thirdly, it's important to really share this taxonomic knowledge with the local people who are the custodians of biodiversity. And at RBGE, we've been working with local communities in Nepal to take the taxonomic data in our databases and present it in a form that puts this information at their fingertips. And the illustration here is the bilingual image-rich plants and new species pages, which were co-developed with the local people and provided very useful in addressing problems of invasive plants and sustainable use of medicinal plants in Nepal. Lastly, if I may, just a couple of comments about funding and lessons learned. The funding of these activities comes from various sources, but it's important to mention that many of these are self-supported through core funding as institutional strategic priorities are aligned with the HE targets and particularly the botanical targets of the Global Strategy of Plant Conservation. For our work in Nepal, we've received funding from the UK's Darwin Initiative and the Department of International Development, now UK Aid. The taxonomic impediment, which features very prominently in Nepal's own National Biodiversity Action Plan, has stimulated government support in Nepal for these projects and addressing the capacity building issues. And I'm pleased to report the government of Nepal is now increasing its funding of taxonomy training and research. There's still much to be done in Nepal, but the knowledge gap and generation gap in taxonomic expertise is now being closed. One of the major lessons learned from my perspective is that all this capacity building and building of taxonomic infrastructure has to be a long-term commitment, so you don't just go in there and do the training. You have to see this as a long-term commitment. Thank you.
Thank you very much, Mark. Excellent and really encouraging. Up to the minute, we are now to the end of today's session. However, please bear with us for 2-3 more minutes just to be able to conclude. Let me briefly check with the Secretariat. I believe I have not, or we have not missed anybody on the list of assigned interventions. So we have concluded successfully that item. Thank you very much to all contributors and speakers. I apologize for not allowing, or at least not facilitating, any discussion. This is really just due to the shortage of time and, of course, the technical challenges we still face. I think what today or this agenda item has shown that there is a great interest and certainly many opportunities to continue with an improved and renewed GTI under the next 10 years of the CBD. So I would like to thank all of those delegates and experts, both from parties and stakeholder organizations, who shared their experiences. We will hopefully be able to capture all of those also when we start the work on the information document, which will happen tomorrow. I trust you have all, this is for the invited or nominated regional representatives and GTI focal points you have received from the Secretariat by email a latest update, I believe, depending on your time zone, yesterday or some half a day ago, about the draft document, which again is just an information document the Secretariat offers to produce under its technical series. And this is in essence what will be the core work part of our meeting for those of you who have been nominated and are invited as observers tomorrow.